Hello students, welcome to EPG Pathshala. I am Dr. Imtiaz Hassan from Jamia Millia Islamia. Today we are going to discuss about module Chemiluminescence and Bioluminescence from paper Techniques used in Molecular Biophysics 2. After this module, a student will be able to understand mechanism of chemiluminescence reaction. Then we talk about the principal application of chemiluminescence, then mechanism of bioluminescence reaction, practical application of bioluminescence, and then we talks about the molecular biology advantage of bioluminescence and bioluminescence imaging in detail. So let's first understand the concept of luminescence. The emission of radiation which occur during returning of excited electron to the ground state. This termed as fluorescence, and in which the excitation is caused by the absorption. But we prefer luminescence because of the high sensitivity. It is a 3 order of magnitude at higher compared to the absorption spectroscopy and linear with the large concentration ranges. So in the luminescence, a spontaneous emission of radiation from an electronically excited species or from a vibrationally excited species, not in thermal equilibrium with its environment. So the chemiluminescence excitation is caused by chemical reactions. The limiting factors is the enormous sensitivity implies purity of analyte or prior purification. Only few molecules exhibit luminescence compared to those which absorb UV or visible electromagnetic radiation. What is molecular luminescence? It is a molecular luminescence is the either chemiluminescence, phosphorescence or fluorescence, in which excitation resulting from a chemical reaction and excitation by the absorption of the photon. So in the chemiluminescence, the excitation resulted from the chemical reaction excited by absorption of the photon, while in the fluorescence it is a kind of photoluminescence. So the basic principle is that the molecules are excited outer shell electrons like in absorbance phenomena, and second, excited species give an emission spectrum that provide information on the quantitation and quantification. So the basic difference between the luminescence and fluorescence is the fluorescence is basically short-lived, while the luminescence ceasing almost immediately. Short-lived with incumbents to luminescence, and phosphorescence features luminescence from 10 to the power minus 4 to several seconds. So simply the fluorescence is the short life process and the phosphorescence is the long life phenomena. Now we discuss about the types of luminescence. Luminescence are of many types depending upon how the luminescence phenomena is generated. The first is the photoluminescence. It is based on the photo excitation, like the fluorescence, that is the spin multiplicity retained, and the phosphorescence, it is the spin multiplicity changed or forbidden. The second one is the chemiluminescence because of the chemical reaction. It is discovered by Ilhard Weidmann in 1888. The third one is the electroluminescence, which is based on the electrochemical reaction, while the bioluminescence is based on the living organism like firefly. Then electroluminescence are because of the electric currents, radioluminescence because of the ionizing radiation, and sonoluminescence because of the cavitation. The thermoluminescence is the re-emission of absorbed radiation upon the heating. And the last one is the mechanoluminescence. It is mediated because of the mechanical action. It is again of 3 types, like the triboluminescence, that a breakage of chemical bonds, fractoluminescence because of the crystal fracture, and piezoluminescence because of the deformation. And the last one is the liololuminescence. This is the dissolution because of dissolution of irradiated solids. Here we discuss the chemiluminescence. especially about the luminol. This is considered as a magic. So the luminol is actually 3-amino hydrazide is an organic compound when oxidized emits light, a phenomenon known as chemiluminescence. Luminol can be oxidized by oxidizing agents such as hydrogen peroxide, hypochlorite, halogen, ozone, persulfate, etc., and generates luminescence signal at 425 nanometer. You can see here the emission wavelength, emission spectra of the luminol. The most significant oxidizing agent of luminol is hydrogen peroxide. In the presence of catalyst, luminol can be oxidized by enzymes such as horseradish peroxidase, lactoperoxidase, and myeloperoxidase. Luminol, you can see here the color of changes of the luminol in the oxidized form. So earlier you can see the luminol and simple luminol and peroxide before adding the hydrogen peroxide, but once the hydrogen peroxide is added, then you can see a dramatic change in the color, and that gives the chemiluminescence signal. Luminol can also be used to detect blood at a crime scene. A mixture of luminol plus oxidizing agent plus alkaline agent, when placed in contact of blood, it emits light because blood containing heme, and it form a reaction and produce a color when it is attached to the light. And that is the major application of luminol in the chemiluminescence. Here we discuss the practical approaches of luminol in protein detection. First, the protein is separated within an acrylamide gel under electrophoresis and then transferred to the membrane. The membrane containing the protein is then incubated with a specific primary antibodies which can bind to their specific target as shown in figure. Horseradish peroxidase enzyme conjugated secondary antibodies which bind to the primary antibodies helps in the detection of desired protein. And HRP catalyzes luminol in the presence of hydrogen peroxide onto excited state reaction product, and upon its spontaneous decay to its ground state, this product emits photon which can be detected by film or sensitive camera. The other practical application of luminol in the nucleic acid detection. You can see here in the figure, a biotin labeled DNA binding probe is designed and immobilized on a membrane containing target DNA sequences. A streptavidin tagged with the horseradish peroxidase then used to bind to the DNA probe complex, streptavidin biotin complex formation in the presence of complementary sequences. Bioluminescence is emitted from the reaction of luminol in the presence of oxidizing agent by HRP enzyme which lead to the light emission. Now we discuss the bioluminescence. Bioluminescence is a natural phenomena of light emission conferred by the living system. The process is caused by an enzyme catalyzed chemiluminescence reaction. The light product by bioluminescence is cold light emission, where the heat production during process is negligible. Approximately 70% of marine organisms exhibit the phenomena of bioluminescence for various biological purposes. Bioluminescence plays an important role in ecology of the ocean. Firefly is the most common terrestrial bioluminescence organism. Variety of firefly species are found in temperate and tropical region. You can see here some of the organisms showing bioluminescence. Now we discuss about the bioluminescence or the action of luciferin and luciferase. You can see here in the figure that bioluminescence is mainly comprised of two main components, a light emitting substance known as luciferin and an enzyme that called luciferase which catalyzes the conversion of oxidation of luciferin into the oxyluciferin. The luciferin reacts with the ATP to create a new compound. This new compound then reacts with the luciferase which adds the oxygen from the and creating a new compound that decay quickly by emitting photon of the light, that's the oxyluciferin. Different species of bioluminescent organism produces different luciferase enzyme. For example, 5' luciferase, Renilia luciferase, luciferase from shrimps, and bacterial luciferase, which have different shapes and therefore different color emission. Here we try to show the luciferase activity in the bioluminescence. You can see here that the luciferin luciferase reaction in firefly. Light emitting reaction requires D-luciferin, ATP and oxygen. A luciferase and a cofactor magnesium catalyze the reaction to generate oxyluciferin, AMP, inorganic phosphate, carbon dioxide and light. Below we are showing the spectrum of visible light. White light consists of light with the wide range of wavelength, and photons related to the shorter wavelength, violet and blue, have higher energy than the longer wavelength. So the oxyluciferin is simply detected and observed in the bioluminescence. Here we show the structure of luciferase. The luciferase in an enzyme, 550 residue long enzyme, and present in the Photonus spiralis and eastern firefly. It having 4 different types of the domain. You can see C-terminal domain, C subdomain, A subdomain. Oxyluciferin is present in the inner core and B domain. During the enzyme catalysis, the small C-terminal domains come closer to a cavity in N-terminal domain, where the substrate D-luciferin and ATP can be accommodated and forming the active site. Here we discuss the biological application of bioluminescence. It is used for in vivo and in vitro imaging. Bioluminescence is used for protein detection and co-localization. It can be used for diagnostic and proteomics purposes. Single biomolecule imaging. It can be used for the gene expression and promoter activity. In the Western blot, bioluminescence can be used. One of the most important technique that is based on the bioluminescence is known as bioluminescence resonance energy transfer. Later we discuss in detail about the BRET. It is used for the detection of contaminants and visualization of protein binding and of course protein folding. A large number of luciferase HRP conjugate system is used for the antibodies detection as immunoassay for the infectious disease test, adsorbent bacterial detection in the cell trapping, fluorescence labeling for the analysis, DNA analysis of the fluorescence, and proteome analysis, and DNA probe where SNP analysis and gene diagnostic tests. Here we compare the bioluminescence with the fluorescence. So bioluminescence is emitted from the reaction of luciferase enzymes and its substrate. Example, firefly luciferase and luciferin respectively. However, fluorescence is the product of fluorophore, for example FITC and Alexa fluoride, absorbing the energy from the light source and emitting the light energy at the different wavelength. Now we discuss about the practical approaches of the bioluminescence. It is used in the estimation of promoter activity. The bioluminescence assay have become increasingly used in the biology for and medical research in addition to the chemiluminescence and fluorescence. The luciferase enzyme isolated from the different species have inherent variability in light emission, allowing 2 or more luciferase enzymes to be used in combination. A genetic regulatory element placed upstream of the luciferase gene and its transfer in the plant and animal cells to detect the system. And the expression of luciferase reporter gene is then measured to quantify the activity of regulatory element or protein in the biological pathways. Here we discuss about the types of luciferase reporter assay. There are 4 different types of luciferase reporter assay. The first one is the single luciferase reporter assay. You can see here in the figure, after transfection of the clone containing luciferase gene driven by regulatory promoter, cells are lysed in appropriate buffer and lysate are prepared. Cell lysate then treated with luciferin supplemented with metallic cation. The amount of expressed luciferase protein can be measured as a factor of transcriptional activity using Lumidimeter. So this way we can assay the reporter gene. Here we discuss about the dual luciferase reporter assay. You can see here in the figure, firefly luciferase protein is used to produce a yellow-green light after catalysis of firefly luciferin. Ranilla luciferin protein produces blue light after catalysis of ranilla luciferin cylindrazine. The light intensities from the dual reporter indicate the promoter activities in the living system, and the target promoter activity is normalized by the control promoter activity through right intensities. This is the third one. Luciferase reporter assay is the dual secreted luciferase reporter assay. You can see in the figure that the secreted luciferase C-Luc and MET-Luc and G-Luc are cloned from the luminous organism Ostracode, Cyprinano, Toluca, and Capinpods, Maleta longata, and Gausia pinceps. After transfection into the mammalian cells, these expressed luciferase secreted into the medium via the endoplasmic reticulum and the Golgi complex. Cellular lysis is not required. Promoter activity can be measured in a real-time way. This kind of reporter assay is best suitable for the high-throughput screening of expression of multiple genes. At last, the multicolor luciferase reporter assay. The multicolor luciferase reporter assay reduces the amount of sample needed and ease the data interpretation. Beetle luciferases such as CBG luciferase, CBR luciferase, E luciferase, S luciferase, SLR, SLG, and SLO from the several species are used, which emit different colors on reacting with firefly, which can be separated by an optical filter. And then multicolor reporter assay is the best suitable for the high throughput screening of the expression of multiple genes. You can see here in the figure how this assay is performed. It's simply showing the bioluminescence phenomena where the different types of luciferase produce different types of color and then can be used to estimate the different kind of biological activity. Here we discuss some important application of bioluminescence. You can see here the reporter gene of bioluminescent organism identified and cloned in the appropriate vector that can be used in vitro and in vivo for the detection of biological activities. Simple imaging system is the detection process, and high and stable sensitive detection methods are available which can be helpful for the data analysis. You can see here in the bacterial cell, mammalian cell, the bioluminescent reporter genes can be incorporated. Then the animal models can be produced by implanting, and then the data can be easily taken by imaging. Here we discuss about the bioluminescence imaging. It is a breakthrough in the imaging system of the living organism. You can see here that the researchers have developed a technique to locate cancer cells by synthesizing a near IR emitting protein and then conjugated it with a therapeutic antibody to produce a probe of cancer cell. This fluorescent dye emits near IR light when energy transferred in vivo chemical reactions. Because near IR light can easily penetrate living system, it can be observed externally. And liver cancerous cells implanted in a mouse can be visualized and tracked using light emitting probe with CCD camera. Therapeutic protein conjugated with a luminescent probe could be detected for a spatial distribution and proliferation of cancer cells. You can see the amount of cancer cells can easily detected in the mice. This is the another example of bioluminescence imaging, which is used for the visualization of drug intervention. Therapeutic drug against target protein could be tested directly in vivo using bioluminescence imaging. Briefly, cancerous cells implanted in the body producing a specific antigen, Luciferase labeled antibody, then subjected to produce luminescence. Drug in the defined dose then administered into the model organism, and imaging done at prescribed times to see the effect of drug in real time. You can see here in the figure that drug administration in mice led to substantial decrease in the proliferation and growth of tumor cells, as indicated by light emission captured in near IR camera. So, as the drug gives its response, the intensity of tumor cells is going down. Unlike BRET is initiated by an enzymatic reaction and thus does not require any light excitation, and resulting in an excellent signal and greater sensitivity. In addition, FRET-based leads to the phototoxicity and photobleaching. The BRET assay involves protein of interest fused with either donor molecule such as renilla luciferase or an acceptor molecule such as green fluorescence protein or enhanced yellow fluorescent protein. So BRET fusion proteins are generated by engineered cDNA from both the protein of interest, donor tagged and acceptor tagged construct are co-transfected into the host cell. The presence of energy transfer between the donor and acceptor molecules then measured and the amount of energy transfer correlates with the extent to which the specific tagged molecule exists within the proximity of each cells. Here we talk about the bioluminescence resonance energy transfer method. You can see here easily in the figure that BRET assay has been developed to study protein-protein interaction. Fusion protein from the renilla luciferase and yellow fluorescent protein are co-expressed, and luminescent signals are measured at 480 nanometer for the renilla luciferase and 530 nanometer for the yellow fluorescent protein upon addition of renilla luciferase substrate cilantrazine. If the protein does not interact with B, if A protein does not interact with the B, and if renilla luciferase and yellow fluorescent protein are not at a BRET permissive distance and orientation, non-radiative light emission is mainly measured at 480 nanometer. However, if A protein is in close proximity or interact with the protein B, placing renilla luciferase and yellow fluorescent protein at a BRET permissive distance less than 100 angstrom and orientation, non-radiative energy transfer can be measured at an increased light. So these two things can be used to detect for the estimation of protein-protein ligand interaction, where we can easily measure that whether the two proteins are interacting or not in a particular biological process. Here we discuss the advancement of the BRET assay. The development of nanoluciferase, which produces an intense and sustained luminescent signal, allows the accurate quantification of very small number of entities. Nanoluciferase is a 19.1 kilodalton ATP dependent luciferase that utilizes a novel cilantrazine analog known as furimazine to produce high sensitivity, high intensity glow type luminescence. Nanoluciferase produce an intense luminescence allows the detection of very small number of entities. Nanoluciferase was cloned from the cDNA isolated from the deep sea shrimps, utilizes a novel cilantrazine analog to produce high intensity glow type luminescence. Nanoluciferase can also be secreted from the cells and allows kinetic studies on the same cells without cell lysis. Now we talk about the advancement in the BRET methods. So the development of nanoluciferase from the deep sea shrimps have improved the luminescence expression in the mammalian cells around 2.5 million fold by merging optimization of protein structure with the development of a novel imidazopyrizonone substrate that's known as fluoramazine. The nanoluciferase produces luminescence with a half-life more than 2 hours and a specific activity is about more than 150-fold greater than that of either firefly luciferase or renilla luciferase. In fact, the nanoluciferase enzyme exhibit high physical stability and retaining activity with the incubation up to 55 degrees Celsius in culture medium for more than 15 hours at 37 degrees Celsius. Here we show the different types of luciferin substrates of the substrates used for the BRET. For example, the beta luciferin, which is converted into the oxyluciferin by using firefly luciferase in the presence of magnesium. The another is the cilantrazine. It is converted into cilantramide by renilla luciferase. And the third one is the fumirazine. It is oxidized by nanoluciferase and converted into fumaramide. In all the three cases, it emits a high intensity of the light and which can be detected in the forms of luminescence signal, and you can say the concentration of log of this substrate with the luminescent signal and a standard curve can be obtained for the all three luciferins and used for a comparison of sensitivity and brightness in the different types of luciferase assay. Here we discuss about the analysis of biological process through the protein labeling using the which is based on the chemiluminescence. The protein labeling can be done for the gene expression analysis, whole genome transcriptomics, fluorescence imaging, protein folding study, molecular imaging, immunoassays, cell metabolomics, macromolecular interactions, and of course a large number of industrial applications. Diverse biological studies are requiring a specific and sensitive approach for more precise strategy to label and visualize protein of interest. Protein labeling represents an attractive technique that can be achieved by basic molecular biology approaches, yet giving extraordinary advances. High specificity can be achieved by bioluminescent protein labeling, which shows the unique advantage for studying protein trafficking, conformational changes, dynamics of protein-protein interaction, and the alteration of protein folding or assembly status. Protein bioluminescence tagging not only provide advantage in molecular biology but also helps in various industrial applications. So students, let us summarize that what we have learned in this module. Chemiluminescence and bioluminescence are common, extremely versatile and useful analytical tools. Bioluminescence is a natural phenomena in which microorganisms produce and emit light via an enzyme-mediated biochemical process. The luciferase is a general name applied to the class of enzymes that are responsible for the converting chemical energy into the light energy. The substrate molecule upon which the luciferase acts are called luciferin. Both luciferase and luciferin structure have and their respective reactions are widely used between the different organisms, and requirement of atmospheric oxygen for the bioluminescence activity remain universal for all the various systems. The main purpose of the macromolecule conjugation is the reliable detection, localization, detection of protein modification, isoforms in multiplex sample, and quantification of the compound of interest along with the real-time monitoring of biological process. A considerable enhancement of the detection system through development Developed techniques have led to the enhanced sensitivity and simplification of the detection workflows. The protein conjugation techniques are extensively and routinely being used to monitor the presence of marker proteins trafficking through the cellular organelle, multimeric assembly, and conformational changes. The attached reporter group label is often required for the detection and biochemical cellular characterization of protein and binding parameters. The visualization of cell and cellular structure as well as tracking of the molecules such as nucleic acid and protein in living cells is mandatory to identify, map, and analyze the underlying biochemical process in the cell signaling. Thank you.